vegetative propagation has some advantages as well as some disadvantages. What are the advantages? By vegetative propagation, you can get genetically identical plants, what are described as pure lines. Another advantage is plants can be developed in large number. One more advantage is time required is very less compared to natural processes of reproduction. So genetically identical plants, large number can be obtained in a less period of time. Therefore, vegetative propagation is particularly practiced for producing seedless varieties of fruit plants. Seedless varieties of fruit plants. But there are some disadvantages. The greatest disadvantage is no variations. Since they are genetically identical, there will be no variations. So in the absence of variation, there will be less adaptability of these plants. Propagules, whether it is the root or the stem or the leaf, whatever it is, they cannot be stored for a long time. Like what is done in agriculture, the seeds can be stored, but the propagules cannot be stored for a long time. Yet another disadvantage is if the parent plant is affected with a disease, infected with any pathogen, the same disease can express in the offspring also. And one more disadvantage is there is no chance of dispersal in vegetative propagation. Where the parent plant is there, in the same region the offspring are going to develop, so there is a possibility of overcrowding. In addition to these methods of vegetative propagation, in the recent years we are hearing about one more method of vegetative propagation, which is called micropropagation. Micropropagation, also called tissue culture. It is a method where any vegetative part of a plant, a portion of a tissue or a portion of a stem or a leaf, can be removed from the plant. What is removed from the plant is called explant, and this explant is grown under artificial conditions, aseptic conditions in the laboratory. So in vitro methods giving rise to tiny plantlets, and these plantlets can be put in the soil and can be allowed to develop into new plants. In this method, since the entire process is being carried out under aseptic laboratory conditions, the resulting plants will be disease free and they can be obtained in large numbers. That is why this process is known as micro propagation.